Hello, everyone. Welcome to day 2 of CCNS. We are going to start today with our trainee spotlight, our second edition of this. We're going to kick things off with Gabriel Benino, and we'll have Evan go after him since we had a bit of a delay. So, Gabriel, go ahead and kick us off.
Well, thanks a lot, Scott. So hi, everyone. So firstly, I'd like to thank the committee for the opportunity to speak today. So today I'm going to talk to you about the dynamics of topographic spiking network models on scales ranging from hundreds of microns to many millimeters. So the first goal of our present study is to understand whether spontaneous traveling waves occur in spiking network models with biologically realistic connectivity during the asynchronous irregular regime. So this is an important question because while traveling waves emerge quite generally in small networks with reduced connectivity, it remains unclear whether traveling waves can coexist with the low rate, highly irregular, low correlation spiking regime consistent with the cortical background state in vivo. And so the second goal is to understand whether these waves affect sensory computations in cortex. So in neuroscience, we're often interested in the relationship between local populations and the microscopic spiking activity of single neurons. Plotted here is the LFP, or local field potential, for a group of cells, as well as the spiking activity of a single neuron in that region in our computational model. So spiking activity is dynamically modulated by the instantaneous state of the local network, which is in turn reflected in the LFP. Now, instead of plotting the LFP only in time, we can plot it as a movie where it now appears as a dot taking some kind of random walk at each point in time. This is often how we think of the LFP, as a kind of noise process with some degree of correlation across space. Now, if we add multiple points across space on the 100 micron scale, we can see correlated fluctuations begin to appear. And it's now apparent that there's some spatial correlation of the LFP across typical distances you'll see between two electrodes in an experiment. Now, if we zoom out to an even larger scale, we see that there are clear, coherent spatial temporal patterns apparent. This is a spiking network model of 1 million neurons on a sheet of 6 millimeter by 6 millimeters, with the LFP calculated at each point in space. And today I'm going to tell you that spontaneous traveling waves emerge in the self-sustained asynchronous irregular activity of spiking networks due to axonal conduction delays. Here we study a large-scale spiking network model with excitatory and inhibitory neurons operating in the balanced conductance regime arranged on a two-dimensional sheet wrapped into a torus. So connections in the network are local and random, with connection probability from neuron I to neuron J being Gaussian as a function of distance, and axonal time delay growing linearly with distance, where this term here, V sub c, is the axonal conduction speed, set to match the unmyelinated fibers in layer 2-3. So we study this network as it scales from 10,000 to 1 million neurons on sheets with side lengths ranging from 0.5 millimeter to 6 millimeters, always keeping the same neuronal density at each scale. Importantly, the networks have connectivities ranging from 100 to 3,000 synapses per neuron, consistent with the dense connectivity in cortex. And this means that individual synapses can be weaker than in networks with reduced connectivity. So this work was made possible by a set of specialized efficient C code called NetSim, which will be released along with this work. So we start first by simulating self-sustained activity in random networks with a constant homogeneous synaptic delay. And we also show this activity, but for the topographic network, which includes local connectivity and linear axonal conduction delays. And at this point, it's actually worth noting that the activity in vivo data looks actually quite a lot like the topographic network model. And so I'll return to this shortly. So now, although the random and topographic network models qualitatively look similar, their distributions of mean firing rate, qualitatively look different their distributions of mean firing rate and coefficient of variation are statistically equivalent, and both within the asynchronous irregular regime. And this result indicates that AI dynamics generated by the familiar random network are preserved in the proposed topographic network. This correspondence in these statistics may be counterintuitive, as these waves may seem to induce strong spatial correlations. But the waves in our model are sparse, meaning that a very small proportion of the neurons contribute to any given wave. This is in contrast to smaller scale models, which have stronger synapses and result in denser waves. Furthermore, the wave dynamics in our model generalize for many pairs of excitatory and inhibitory conductance. So within this black contour are all the realizations of AI dynamics, and wherever there's a colored square is where waves were detected. Now, if we focus on this lower region with lower values of these two parameters, we actually see that the waves are strongest here. And this is consistent with the conductance states, or the conductance regimes observed in vivo. So with this first question answered of whether spontaneous traveling waves can be generated in a realistic model that obeys the AI dynamics, we now shift our attention to the question of whether these waves affect sensory computations in cortex. So a good place to start would be turning to the biological data. So spontaneous traveling waves are found to robustly occur in the multi-unit LFP recordings in primate visual cortex. And these waves modulate perceptual sensitivity. In visual detection tasks performed by the implanted primates, the success rate was found to be significantly higher when the waves aligned a negative LFP with the incoming sensory-driven spikes. So from these results, it's clear that these waves can serve to boost sensory computation depending on the phase of the wave. So I could perform a similar test in the model in order to understand how waves modulate external input in a phase-dependent fashion. So this test used 3 identical runs of the same network, first with no stimulus applied. So we see this red line here of the rate of a local group of excitatory neurons, and the light blue line shows the corresponding LFP. And we see there's a counterphase relationship. So wherever the LFP is high, the rate is low, and vice versa. Now, if I apply a stimulus during some period of positive LFP, the rate increase due to the stimulus is actually quite small. Now, if I apply the same stimulus, but during a period of negative LFP, the rate increase is much larger. So by having a good model for the dynamics of neural populations, we can now study in detail what kind of computations result. So in summary, traveling waves robustly emerge in the ongoing AI state due to axonal time delays. This is observed both in our topographic network model and the in vivo data. Furthermore, these waves modulate perceptual sensitivity depending on the phase of the wave. For example, in the primate experiments, visual perception was increased when the wave was aligned with the visual stimulus, and in our model, the local stimulus evoked firing rate is either suppressed or boosted depending on if the stimulus is at a depolarized or positive or hyperpolarized or negative phase, respectively, of the LFP wave. And so in closing, I'd like to say thanks to everyone, and I'll be happy to take questions.
Awesome. Thank you, sir. So one quick question and one longer question. John was curious about what actual model you were using for the spiking neurons.
Great. Thank you for that question. So we're using leaky integrated fire model for the individual neurons.
Perfect. And then another John had the question, do the periodic toroidal boundary conditions have any influence on the model behavior?
So only in the sense that the wave might go to the edge of the square that I showed and then come back on the other end. Yeah, so it just appears like periodic waves.
Fantastic.
Yeah.
So basically, they don't bounce off; they sort of cycle back around.
Yeah, that's right. And the reason why we want to have these boundary conditions is for the connectivity itself. Otherwise, if we had just like a sheet with edges, then the synapse, the connectivity at the edges of the network would be different than in the center of the network.
Gotcha. Awesome. Thank you very much, sir. I am now going to somewhat unceremoniously kick you
Yeah,
off
go for it.
screen. And we will get Evan up here. So Evan, go ahead and share your screen. Evan will be our second trainee spotlight of the day. So whenever your screen is up and running, which it is now.
You see it?
Yep, you're good to go. So take it away, sir.
All right, thank you. And also thank you to the organizers for putting together really a great conference. I want to wish everybody a happy morning. And my name is Evan Cresswell-Clay, and I'm going to be presenting on some work from my collaboration with Maurizio De Pita, who some of you will have seen, or you will have seen yesterday in the beginning opening session. And I'm going to be discussing specifically astrocyte morphology. And astrocytes are a cell that Maurizio actually had mentioned in his talk and discussed in his talk, and we're going to be exploring them further in this presentation. So astrocytes are a type of glial cell which are found throughout the brain, in fact, everywhere in the brain. And as we can see here, we have a readout of calcium signaling. And while neurons respond to stimuli via electrical signals, astrocytes respond to the stimuli via calcium signals, as we can see in this experiment where a mouse is presented some type of visual stimulus, a kind of oriented grating, and they record from the calcium signals from a single astrocyte in the primary visual cortex. And from these rows of calcium traces in these different regions within the cell, we can kind of see a segregation of these calcium dynamics in this single cell. And we want to understand what causes this segregation. And one way to explain this is by looking kind of at this mapping here of the calcium dynamics in another cell, and seeing that, again, we have kind of these hot spots of calcium activity spotted throughout the morphology. And these hot spots correspond to these subcellular domains, these microdomains of calcium activity, which are showing variegation showing variegation both spatially and temporally. And this leads us to our question, which is, how does the morphology of the cell interact with these kind of spatio-temporal variegation in these calcium microdomains? This is not an easy question. So if we look at this beautiful reconstruction here of an astrocyte, we can see that it's characterized by many processes and bifurcations and shows a pretty severe degree of complexity. And we want to try to understand and characterize this cell based off of its morphology. So in order to do this, we're going to be using 72 cell morphologies, which were taken from immunofluorescence staining with Clovrel and her collaborators in her 2019 paper, where they were recording from a mouse cerebral cortex looking at astrocyte structures. And from these structures, they were able to digitize specifically the primary and secondary, the branching structures of these astrocytes. So given these morphologies, we now want to use and develop a principle of these astrocytic branching structures. So if we have one of these branching structures, this specific morphology here, we can extract from it several, all of the potential anatomical variables which would describe its morphology, such as height or tortuosity. And from these variables, we get a high-dimensional morphological space that represents an individual cell, a point where a point in that space represents an individual cell. And across our 72 cells, we want to then simplify this high-dimensional space into a kind of a lower-dimensional version of only choosing variables which are expressing multimodality, multiple peaks in the distribution across our 72 cells. So given this morphological space, we can then use unsupervised clustering to organize our 72 cells, our group of cells, into 3 groups. And we can show, we want to show that these groups are showing this type of stratification in morphology across that morphological space. So we can resort to linear discriminant analysis, where here we see a 2-dimensional embedding in in the variables of that analysis, showing clear separation of our three clusters. And as well, we can focus on the individual components of morphology and again see the separation across our three groups of cells, specifically in the number of branches or tortuosity or cell surface area. And from this, we want to do some further analysis to understand these the expression of these groups. Specifically, one type of analysis is shoal analysis, and we see from this analysis in the middle that we have, as we move out from the soma, if we plot the number of branches in a given cell, we can then see these curves representing the branching morphology as you increase that radial distance from the soma. And we see a stratification, again, of our three clusters across this analysis, further showing the clear separation of our 72 astrocyte morphologies, which is interesting because, again, to remind you, these cells are all coming from the same brain region, or to reiterate that they're coming from the same brain region. And from these different groups, we now want to, and this stratification, we want to create a generalization of these kind of the morphologies in a specific group by looking at a consensus, a topological consensus. And as we see here, when we create these averaged morphological representation of our three groups, we can see that it's characterized, by specific branching structures such as dense arborization across these three different clusters. And this is the main kind of takeaway to be able to express these 72 cells into three morphologically different groups, generate these consensus topologies, and then move forward to the next episode of this chapter, which is to consider how these different consensus morphologies will reflect in terms of the calcium activity in the cell, and to see whether we can further extract these morphological principles from the astrocyte clustering in order to investigate its response in this calcium activity. Thank you for listening.
Awesome. Thank you very much, Evan. So I have one question, which is I love how you've been looking at astrocyte morphology because we're learning more and more from neuronal morphology is very important for how neurons operate. One of the things we know is that one of the biggest differences between human and rodent brains is that there are big differences in neuronal morphologies. Human neurons tend to be larger, etc. Have there been any studies of astrocyte morphology sort of comparing across species, or are we not there yet?
Not to my knowledge. I know that they've looked at the differences between astrocyte morphologies across layers or age within species, but I'm not sure about between species. Although I think there are some general results on like the complexity versus like human versus a lower mammal.
Okay. But basically what they have seen and what your work shows is that there is this sort of correspondence between the astrocyte morphology and its location and sort of the for broadly speaking the role that it's serving. Is that
and this is the main goal was to to try to start to organize these cells even within a specific region of the brain, and to look at the morphology within and characterized by that. Because the idea is that this morphology is linked to and shaping the hierarchy of calcium events that you observe in the cell. Yeah.
Awesome. So if you can answer this really quickly, that'd be great. Dante asked, how can you be sure it's three very distinct clusters and not a bigger Gaussian of which you're taking three clusters, like one mean, one high standard deviation, one low standard deviation?
Yeah. So that's where the discriminant analysis comes in to show, to see whether or not we are getting a higher some level of stratification. So, and I think within the individual variables, you sometimes do see that we're just looking at different distributions within a single Gaussian. But when you look at when you look at one variable, that could definitely be the case. But when you take a combined kind of snapshot of the morphology and you start to consider it in this higher dimensional space, you can see the grouping of these cells, the very specific grouping of these cells within that kind of canonical that linear combination of variables.
Fantastic. Thank you very much, Evan. So at this point, we're going to start the 3 parallel sessions that we have. So I would encourage everyone to go to the parallel session that interests them the most. Again, if you click the schedule tab on the top left of your screen, you'll be able to scroll down and choose which of the 3 talks you'd like to go to. So yeah, please go enjoy those fantastic trainee talks. And thank you to Evan and Gabriel for these fantastic spotlight talks. Bye all.
Thank you very much.